Very good morning, everyone. And yesterday we were discussing about several techniques that are involved in verifying or trying to figure out the genetic variations. So we did discuss about several types, including about little bit biology of transposons. We discussed about what is meant by target site duplication, and that is a common feature, which we will discuss again later. And to understand this, we have seen the simplistic explanations as well as a little bit advanced in that sense about how to be open when we are looking into results, and how fast we arrive is important. Speed is important, but because this is about science and it is philosophy, it is about truth, not necessarily coming to conclusions fast, but coming to conclusions accurately is very important. So while the priority goes to accuracy, when we are accurate, we want to be accurate. The best possible way or the approach is to be as open as possible, see all possibilities, and then figure out the most probable one. When we say most probable one, the approach in science is like this. Say, for example, there are 10, or say there are 4 answers for a question, or there are 4 possibilities, and you want to have assume that there is only one correct answer out of the so many that is given. Do you pick the correct one out of these 4, or there is other this is something like picking the answer, right? Cherry picking. And when we are picking, it is we doing it, so we don't know our biases properly, we don't know how we think sometimes, and we might be sometimes wrong too when we are picking. The approach in science is also about this, by process of elimination, right? That means you eliminate out all the wrong answers, or most improbable ones, you remove them out, and what is left is supposed to be the correct answer or most likely answer. But then there is a problem, of course, that we cannot be sure. When we don't consider our biases, and that the possibility that we might have made a mistake, then we are very sure, because our mind doesn't take into account all these things about us. The mind thinks it is right. Mind is us and us. But anyway, it's just for sake of explanation. Is our mind is filled with biases, or our needs, our urgencies and priorities. And based on that, if we are going by picking, we might pick something, we may pick something wrong. So the efficiency as a scientist will come in understanding self. Of course, you need the drive to work, you need the grit or perseverance, you need to have a big heart, in the sense that even though you're failing, you still have to keep moving ahead, perseverance. And these are all important things. But the other important thing is to understand self. What kind of mistakes do we make? What kind of biases do we have? And that is an important part of critical thinking. Okay, so I would like that you practice what I'm also trying to say in this case. We could just say, yeah, there is one copy here, there are 2 copies here, and move away. But that's not what we should do. Our job is to think, and that is what is a scientist's job. Think accurately, think efficiently. with speed and priority as, and the priority is accuracy, because we don't want, science is about generating knowledge. And if we are not accurate, we might generate something faulty, and that is anti knowledge. So we don't want that. Okay. Anyway, so we need to approach with the same way in majority of the cases. So that is one of my attempts when I'm trying to explain the results in an altered way, or a deviation from what is too obvious. Then we also learned about VNTR, how to see that, and the case here is about designing the primers, and then based on the length we could identify it. And then I hope you have had a look at the mobility, electrophoretic mobility of DNA, especially those of the covalently closed circular DNA, which are usually supercoiled, which are usually negatively supercoiled. And I hope you know what is meant by positive supercoiling and negative supercoiling. Watson has some animations too. It will be nice if you can read the book or watch the animation online somewhere. So the more supercoiled, it can run faster. The linear one apparently runs second in these three races, and the open circular one travels little slower than the others. Then we discussed about single strand conformation polymorphism. The sequence variation in sequence can cause variation in conformation, and variation in conformation can cause variation in mobility. And that is what that. So if there is something of the same molecular weight, almost same, but they are moving in different rates, that means it is about change in the conformation. And that conformational difference could have yielded from the variation in the sequence, because conformation is based on base pairing, intramolecular base pairing, and therefore we can assume that there is variation. We wouldn't know what is the variation. We would then know that there are variations, and we probably will have to go for something like sequencing. Not many people use this technique anymore, because there are sophisticated ones, or simple things are easier ones are now available. But the important thing why I do like to cover these is about trying to understand and revive the biology that we have learned, and try to make logical connections between biology and as an application or as a tool. So we came to about genome-wide comparisons. We did discuss about different approaches, holistic or reductionist approach, where some people work on the whole system level, some people work on specified genetic factors, and both are important. And why I do tend to side sometimes with reductionist is, since the evolution of humans, we know about our body and everything, and our knowledge level happens to be a little bit like this. But once we have learned, we invented tools and other things. Now the people never knew anything about cells. They knew about organs, right? But once we have learned about cells, once we have learned about subcellular ones, and then we have learned about genes, our knowledge level has taken off in a different level. Okay, I'm just telling you about the approach. Everything is a system. It only depends on what level you are looking at, right? A molecule can be a system too, if you're considering the atoms as components, or even subatomic particles as components. Anyway, I hope you understand that both are important, and this is for several hundred thousand years we are like this, and all of a sudden, when we knew about these things, our knowledge level has transformed dramatically. And then we were discussing about the genome-wide comparisons, where you take the whole genome and then check. If I didn't mention, it will also be nice if you can go to and try. This is a karyotyping technique where you can find chromosomal aberrations in much more simpler way. But you cannot be specific details. All you can probably say is this part of the chromosome is missing, and this part of the chromosome has duplicated or translocated to some other chromosome, and so on. It doesn't give fine details about the sequence, but at the level of chromosome, and if there are large scale changes, then karyotyping can help. So we were discussing about the microarray technology. Microarray is each dot here. Each dot is is embedded or stuck with several single strand of oligonucleotides. That oligo would have been specific for gene X. This spot could be for gene A, this could be for Y, and this could be for Z. And we have printed all these things just about a very small, something like a glass slide. And we will allow the hybridization. And the hybridization, if in this spot, for example, only the red ones are bound. So when you upon treatment with lasers and fluorescence under the microscope, the computer will detect this spot, this area as red, and this area as green. And if two of the different strands bind, then they would give something like yellow color. So it's about the way we set up the experiment that is important. So as is given here, yes, sorry. So it's all about experiments and the experimental design or how one has chosen. In this, everything will have a test and a reference. Test is the one that you are actually testing, in the sense against a reference. This is something like the control. You know the details about it, what genes are present, what genes are not present, and you're making a relativity. You're comparing the test with the reference. So you need to get DNA molecules, and if it is direct genome, they would fragment the genome with restriction endonucleases, or sometimes with mechanical shearing. They would have broken it down into smaller fragments. And then the labeling. One of it would be labeled with a red fluorophore, and the other one could be labeled with a green one. And then you allow these to hybridize onto the microarray. And once the hybridization is over, you clean it up, wash it up, and then you incident laser light, and then you will see the fluorescence, whether it is red, green, or yellow. So at one point, you may have the laser 1 to excite one fluorophore. At another point, you might have the other one, green fluorophore, to excite the green fluorophore. The computer can make an overlay of, digitally, it will make an overlay of both the pictures. And then if say, for example, this one is showing red, and this one is also showing green with two different fluorophores. And when you merge it up, it shows something like yellow. That means that gene is present in both the test as well as the reference. Okay, so that is how one can figure out the presence or absence of genes in genomes. The same thing, it is about when we say at the genome, and you can also do it at the cDNA. This is more popular, where you do the expression analysis of all the genes. That is the transcriptomics. Transcriptomics is about, what is a transcriptome? It is the total mRNA of a cell that is present at that time and condition and the growth conditions. So, I mean, the principle will remain the same, almost what we have discussed just before. Here, we take the mRNA, we would have made into cDNA, and labeled, mixed, and hybridized onto the chip. And then the analysis, as we have discussed before. So this is where we will talk about analysis of gene expression. We have started somewhere here. We started about gene variation, and we have done a problem using the, I hope you remember, southern blot, northern blot, and western blot. So we will also discuss about northern blot a little bit here. The question that we did a few days back, that's very important. Please ensure that you are aware of it. Okay, so from the DNA, we have the mRNA, and from there, we have the protein. So we need to, as I was describing, the form and function of a cell is determined by these two, the gene, the protein, the proteome, and the transcriptome. How should the cell look? How should the cell behave? And its metabolic activity, everything is determined by these things, which of course are determined by the genome itself. Okay. So if there is a gene X is present in a genome, is there any guarantee that gene X is mRNA X is present? Not necessarily. So and then if mRNA is present, is it sure, are you sure that the protein X is made? Not necessarily. So if gene X is present, it doesn't mean mRNA is present. If mRNA is present, it doesn't mean protein is present, right? But if what we can for sure say is if gene X is absent, then there is no scope for mRNA X. This is not possible. And if mRNA is not gene X is not present and mRNA X is not present, then protein X will also be not present, right? So we still, if from the analysis of genetic variation, we know a particular gene is present in a genome, it doesn't mean it has contributed to the form and function of that cell uh, at that conditions. Say, for example, we have about 210 different cells. We have about 25,000 genes in our protein encoding genes, and we have 210 cell types. Not all the genes are expressed in all. Say, if we talk about an epithelial cell, this epithelial cell will have genes that are required for its metabolism, and it will have expressed the epithelium specific genes and other regulatory genes. The rest of the genes, say for example, genes that have to become the neurons or genes that have to become that of the muscle, all those genes are still present in the genome, but they're all suppressed. They're all silenced. Okay, only these genes are expressed, and that is how the cell type is coming. And we have these 210 cell types. Then this kind of regulation is present in all type of in all these different cell types based on what cell they are. So the presence of gene doesn't mean the expression of the gene. And therefore, we still have to study about the analysis of expression. If there is a gene that is required to make it as an epithelium, but it's not expressed, then it's not useful to the cell. I mean, that is how we need to figure out whether expression is happening or not. So the simplest one is northern blot. We take the total RNA, and when you run the total RNA, you will usually find a smear-like thing, and you will find 2 or 3 distinct bands. These belong to the ribosomal RNA, the bands, because ribosomes are the largest in number. Say, for example, if there is E. coli and it is in its most optimal conditions, in that sense, about 40% of the dry weight of E. coli is just ribosomes, including the proteins and all. That means you have a large amount of these ribosomal rRNA. When you isolate the total RNA and you run the gel, something like this, you will, on the agarose gel, you will be able to view these bands like this, which represent the ribosomal RNA. You may also see one more, the 5S or so band here. And I remember, so if you see these bands, what are these bands? Then some we quickly tend to say that one is mRNA, rRNA, and tRNA. Don't do that. Total RNA, you will be able to see only these bands, and those are that of the ribosomal. You cannot see mRNA as bands because there are several genes produced, and several mRNA are produced of several genes, and each of them are different size and different in number. So some may be very large and few, they will be here, but you cannot, they're not in so many number, so you cannot have it as a single band. Whereas ribosomal mRNA, you can see them like this. Anyway, once that is done, and we then do the blotting onto the filter, and then hybridization with the probe. And in this case, we also have to discuss about the probe. We will discuss now. So the probe can be made up of RNA as well, okay? And it should be randomly labeled. And now assume A is control. What can you say about A, B, C, D? Everything in relation to A. What can we say of A? Anybody? Hello? Okay. So there are two things about northern blotting when I was discussing last time too. You can tell whether it is present or absent. To start with, northern blotting is to identify if a particular RNA sequence is present in the total isolated RNA or not. So the first thing is it can tell about whether it is present or absent, whether whether the particular RNA sequence is present or not. Second thing, it can also say how much. Say, for example, if you have loaded 10 micrograms of total RNA, in all of them, 10 micrograms of total RNA in all of these. And then when we do the blotting and then hybridize, you find A is like this, but B is little lesser. Remember that this is kind of semi-quantitative gene expression, semi-quantitative comparison or technique, you can say. We added equal amount of RNA in each of the samples, but in one it is showing for example, we will say it as we used probe for gene X. In that sense, gene X has a band like this of this intensity that is visible in sample A, whereas in sample B, it is maybe 1/5 or 1/10 of the compared to that of A. What can you say? Does it mean that you loaded less in sample B? No. We loaded equal for all of them. So to start with, it should have been equal in all of the cases, but it's not. So then you can say that B expresses lesser than that of A, or gene X or mRNA X is less in B compared to that of A. That is what you can say. In C, in lane C, there is no band at all. And what does that mean? That means gene X is not expressed in C at all. And in gene D, or sorry, sample D, you have two bands. What does that say? It says that probably you have two different copies of the gene somewhere and also somewhere else, where one of it is a truncated version. So one of it is producing a larger mRNA and the other one is producing a shorter mRNA. Okay. It could also mean that maybe some of the mRNA are degraded, say something like this, and because of which a band is coming. But that's unlikely. Why I say unlikely is we for RNases, there are very few examples which would cut exactly in a sequence specific manner. Not that there are not there. I have in my own research experience, I found there are some enzymes which cleave RNA in a sequence specific manner. Okay, but generally there are not. So the most likely explanation for this is that one of it is a full length RNA, mRNA or RNA, and the other one is a smaller one like this. Okay, and everything we are saying right now is in relation with the control that we have. That is here assuming A. Okay, I hope you understood that part. So before going further, I would want to discuss about the making a probe. This is something very important too in terms of your exam. So I have a vector, something like pgem. If you remember, we had T7 promoter, and then we had, I think, SP6 promoter, and we were able to, there is a multiple cloning site here into which we would have cloned the gene of interest this way. Okay. Now we could, the idea is I want to make RNA probe. How to make a probe. And I want to also see the radio labeling in this. How to do the radio labeling. Anybody wants to try? Which promoter will produce sense mRNA? Which one will produce antisense RNA? It is in this direction, right? And this is the reading of the open reading frame is in this way, right? That means if T7 RNA polymerase was present, it would have recognized the T7 promoter and it would produce RNA that is sense. Right, that is what would have had the open reading frame in this orientation. That means start codon is there and stop codon is here. And that would give rise to a protein, functional protein as encoded. If it was by SP6 promoter, then it would have produced the antisense, that is the minus strand. And it is the antisense. And this will be the plus strand. And this would not be recognized by the ribosome unless there is an open reading frame. This will be the 5' to 3', and reading always happens from 5' to 3'. So this one does not produce any protein. So why am I discussing this is we want to detect in the RNA sample, like we were discussing in this case, where we want to know about the presence or absence of an mRNA, we want to have a probe. And that probe should be complementary to the mRNA. That means the mRNA is the plus strand, and we need to have a minus strand or antisense strand, right? So now, can you tell me which of this should be used or useful? This is what we want to detect, isn't it? mRNA levels are what we want to detect normally in the sample. So we should have produced the antisense strand or the minus strand. So we would have put this vector into a host in which you would have had sp6 RNA polymerase expressed. That sp6 RNA polymerase would bind to this and will allow the transcription of the opposite strand to happen. Therefore, you will get the antisense RNA of antisense for gene X that we want to detect. Okay. There is another possibility. I want you to think about it much more clearly now, because this is an intellectual thing. I love it since the time I learned. And that is how you improvise on the things that you will come across. There are some vectors like this. Both of them are T7. And there are several cloning sites of which within which you might have cloned this one. BAMH1 is here, EcoR1, HIN3, and say KPN1. So how can I produce how can I produce the antisense of mRNA? Say this is gene X that is present, and I want to produce yeah, this is gene X. I want you to produce the antisense RNA for gene X. How can I do that? Anybody? So basically, if I add T7 RNA polymerase, what will happen? Both will produce this T7 will this T7 promoter and the other opposite one, T7 promoter, both will express. And what we will end up is a double-stranded DNA that is complementary. That's not what we want. We want specifically single-strand RNA, antisense RNA for gene X. That means the T7 polymerase that is here should be useful. Only that will produce the antisense. The top one will produce the plus strand, and the bottom one, say the one that is here, will produce the negative strand or minus strand. How can we produce? So some people, what they do is, of course, we are going through in vitro transcription assay, not assay, transcription. In vitro transcription, meaning that you take a tube, something like PCR almost. Only thing now you're doing is you're doing it with RNA polymerase, but it's not a chain reaction either. You give a template with a promoter, you add RNA polymerase, and you add the NTPs, nucleoside triphosphates. You will have the buffers, and anything else is required. I think that's it. So you give this and incubate at 37 degrees centigrade or whatever based on the RNA polymerase and the reaction that should happen. At the end, you will have several copies of the transcripts. Those transcripts can be used as probe. So we'll also look at labeling when we discuss about it. But right now, the idea is how to generate the talk is about the antisense strand. So let's try to look at what would happen if I use EcoR1 and make a cut like this. And now I add in condition B. Okay, previous condition we just added both, and then what will happen. Now in condition B, I have treated with BAMH1, eco R1, sorry. So I have treated with eco R1, and what can you expect? So which promoter will be active, which promoter will not be active? Both could be active. The T7 polymerase that I am indicating now, it will produce the plus strand, no issues. What about the T7 polymerase on the other side? It will produce something, but very small fragment that has nothing to do with gene X, because we are cutting outside that of the gene X that is cloned. So it may produce something, but it's actually not so useful. It's a very tiny fragment as well. So it's not useful. This is not what we want. We want the antisense RNA. So we'll see in condition 3 what would happen. Let me fill this up. Assume that we have the vector cloned, gene X cloned into vector like this, and now you treat it with BAMH1 or KPM1. What would you get? In that case, we have, this is in condition C, we have treated with BAMH1. And if T7 polymerase, the forward one or the first one, it will produce a small fragment which has nothing to do with that of gene X. And the downstream, the other promoter that we have on the opposite side will produce the minus strand, right? And that is the antisense and can be used as the probe. For hybridization, we can use this. So it's not necessarily that we always have a situation like T7 polymerase on one side and sp6 on the other side. We can also have something like this, even T7 on both, but still we can generate the antisense RNA by cutting the vector. There might be some issues with these small fragments, but that's so far, in my knowledge, that is never a problem because they are very tiny compared to the probe. And during the washings that we will do during the hybridization, usually they fall off. So the next question is how to label it, right? How to label it. And labeling can be, we use P32. It's a radio label. And DNA, if you remember Hershey and Chase experiment, no, Avery and McLeod, where you did blender experiment. To label the nucleic acids, we can use P32. To label the proteins, we can use sulfur, right? Because CHNOP, these are common, say, I will just put it again, 1 second. CHO, carbohydrates, lipids, everything has these elements, CHO. N is also present in majority of the lipids, also in special groups and nucleic acids. Phosphates are they are more rich in nucleic acids. And then SC is additional thing that is present. Phosphorus is not present in proteins, but sulfur is present. Right? The choice of which elements or radioactive ones you want to use will depend on what molecule are you looking at and what molecules are unique to that particular, what elements are unique to that particular molecule. So we cannot use CHNOP and CHON or S radionuclides, radioisotopes of those. We can use phosphates, which nucleic acids have it in abundance. So the question is, there are two labeling, you can say random labeling. I'm also covering the other labeling that is called as N labeling. I hope you remember that when we are doing SSCP, sorry to go back, SSCP, we had end labeling here, right? That is called end labeling. In that technique we use, so keep in mind what is meant by it. Random labeling means you have a nucleic acid in both the cases, and random labeling means you would have labeled in different locations randomly. You would have had the radioactive element incorporated. When you say end labeling, it's only either on the 5' side or on the 3' side. Okay. So how can we, for northern blotting, southern blotting, you use random label ones. So let's see how we can get that. I just described in the previous slide about in vitro transcription. We need a template. This is the template, which should have been processed appropriately. We will need to add RNA polymerase. The RNA polymerase that we add will depend on the promoters that the vector has. Say in this case, as you can see, it will be T7 polymerase. But if I gave you a situation like this, it should have been sp6 RNA polymerase. Okay, we should have added that enzyme. And NTPs are nucleoside triphosphates. And for DNTPs is what we will say in terms when we are using it in PCR. Deoxynucleoside triphosphates. And then buffer conditions to allow the reactions to happen. So we want random labeling, and we know that there are phosphates. And if you can see, if you remember the structure of any of the nucleotides, we have, say, this is a base. Okay, we'll put it as there are differences between RNA and DNA. RNA and DNA. If we use phosphate alone, it could label both RNA and DNA. That's not what we want. We only want RNA to be labeled. So as I was telling, in RNA, we will have used NTPs. If it were DNAs, we would have used the substrates as DNTPs. To start with, we are taking NTPs, and the phosphates of it would be labeled, so that only RNA is going to get labeled and never the DNA. And moreover, if we are doing it in vitro and we are going to add only RNA polymerase, this is what will happen. If we are doing it in vivo, then we should have been more careful. And if you want to be even more careful, we say A, T, G, sorry, G, C. I know I made the error is, the difference is that RNA has uracil. And that is one of the differences between DNA, right? So if you look at the differences between DNA and RNA, chemically at the elemental level, okay, we are saying ribose and deoxyribose. I think it is, we'll try to fix it. So this is 1, 2, 3, 4, and 5, CH2OH. And we would have, if there is a base here, nitrogenous base, then we would have labeled 1', 2', 3', 4', and 5', because bases also have numbering 1, 2, 3. To distinguish the numbering of bases from that of the ribose, they put as 1', 2'. So say it as 1', don't say 1 dash. Okay, so the point is, we have OH. This is the others I'm not fixing. So this is ribose. And when we don't have this oxygen, then it is deoxyribose, right? That is, what is the difference then? The difference is 1 oxygen in terms of this ribose deoxyribose. You can also see it as uracil. I hope you can help me if I made a mistake with this. Uracil is something like this. This is 1, 2, 3, 4, and 5. 5-methyl uracil. is thymine. So the DNA, in DNA, there is -1 oxygen atom, and you have this addition. This is thymine. If you have this methyl group, it is thymine. So you can say, and not everywhere, one methyl group plus a methyl group. That is how you can compare with that of RNA, right? I hope you'll remember that. So the chemistry of nucleotide is the bases and the nucleic acids is important for you to do any of the manipulations within E. coli or within the rDNA. And that's why I want you to also have a thorough look at that. Now, during RNA polymerization, what happens is that we have a template. I'm putting it in reverse. And then we have RNA polymerase, a primer is synthesized, prime, and then it will keep on adding. The nucleic acids are coming as NTP, triphosphates. So if I have to put it differently, I will just write it as U, and ribose is there, and 3 phosphates. Okay, this is your uracil. So we have 3 phosphates. This is alpha, beta, and gamma phosphates. And whenever we are using any radioactivity, we want to minimize the amount of radioactivity that we are using. So we should take one of these phosphates should be radio labeled. Which one should it be? Is the question. The point is that we have to have this alpha phosphate be radio labeled, because during polymerization, what happens is this beta and gamma phosphates are released out. They are released, and then they do not get incorporated. They are released out, and they do not get incorporated into the nucleic acid that is being made, the RNA transcript. So we need to ensure that the phosphate that we have, the alpha phosphate is, one second. Yeah, the alpha phosphate is labeled 1, not in beta or gamma. And that would get incorporated there, and that would have been the radio labeled 1. So for this in vitro transcription, we would have used RNA polymerase, T7, for example. We would have used NTP. We would also use something like, say, for example, for this case, I'm just saying it as UTP, but alpha P32 UTP. What does that mean? That means the alpha phosphate of the UTP is radio labeled. So that every time when we have these kinds of things and buffer, RNA polymerase will keep on polymerizing, and wherever UTP is being added, you will get the radio label incorporated into the synthesizing the probe, and that one can be used as a probe. Okay, that is how we label. The random labeling is done, which is used for southern blotting or northern blotting. This is how we want to use. One thing I want you to keep in mind, I'll just take 2 minutes, is that whenever we are using radioactivity, it is dangerous to the health, it is dangerous to the environment. So we want to minimize the amount of radioactivity that we are going to use to start with. Okay, disposing it off is also a lot of it is potential hazards are there. So we want to reduce it. Typically, you could have also taken beta and gamma also radio labeled, but these are not going to get incorporated, and then you have a lot of wastage that you're going to get. So we should have ensured that alpha P32 UTP. That is essential. And the second part that I was saying is to ensure that there are no unnecessary wastage of radioactivity. We don't want exposure. We don't want any wastage from these things. Okay. So that is about random labeling using and probe for northern blots. In the next class, we will discuss more. Any questions? The others may leave if you don't have any questions, but you can ask if you have any questions. Anybody?
For now, no, sir.
Okay.
Like I'll anyway go through those recordings again, and maybe if anything, I'll let you know, sir.
Yes, that will be well. You're welcome to do that. Okay, then. Thank you very much.
Thank you, sir.
Yes, bye bye. Thank you all.